Hello, everyone, and thank you, Pietro and Ricardo, and it's just such a pleasure to be here. What a beautiful city, and it's been already a very inspiring and am I not on? Oh, yeah, it's okay. It's really, and I have to thank Stephen for setting me up because I totally agree. We are on the verge, really, of an incredible advance in medicine driven by genetics. And I want to talk a little bit about, well, first I have to acknowledge the real Nobel Prize winner, which is shown here. I just had to update the slide because this animal just won the Nobel Prize again this past December. Really amazing little animal. It's basically a tiny little human. We shared 3 billion years of common ancestry with this little tiny animal. We are very, very similar to this animal. We have all the same machinery, our neurons, our intestine, on and on and on. The signaling pathways are all the same because we shared this common ancestry. And that has led, of course, to multiple really beautiful fundamental discoveries I don't have time to talk about. It's actually, it's interesting times politically in the United States, as you might say. This is a really fascinating, beautiful animal, but it begins its life as a male making sperm, and then it transitions to making eggs. So we're a little bit worried about our funding, let's put it that way. So it's a transgender animal. Makes it really easy to work on from the genetics perspective. And it's really tiny and simple. So, but I want to zoom in here on the germline of this animal. We call this the germline. This is the lineage that gives rise to the sperm and the egg. This is an immortal lineage. And what you're looking at here is an ovary from this little animal. And the maturing larger cells that you see here are the eggs. And I just love this movie made by my colleague, Jim Priest, because it shows the dynamic nature of this process, and it shows the beauty of it. If you look at the various aspects of the slide, the green, first of all, is truly a stunning feature of the eukaryotic cell. These are called mitochondria. What are they? They are the remnants of a previously free-living organism that became a cohabitant within a primordial ancient cell billions of years ago. And the two organisms merged and became one. That's part of how the eukaryote, what we call the eukaryotic cell, distinguishing it from a bacterial cell, that's part of how it emerged. And then you see the nucleus here, the dark circle. That's where the DNA is. The DNA that's the genomic DNA of the worm, in this case, or the human, resides in the nucleus. But the mitochondria also have DNA. But most of the DNA that used to be running the mitochondrial functions that make, these are like, we call them the powerhouse of the cell. They make ATP, this energetic molecule that fuels the cell. They're little batteries that are functioning kind of like they did as free-living animals. They create energy for the cell. Those guys lost their DNA. Where did it go? There's only a few genes left in the mitochondria. They moved horizontally into the nuclear genome. Information flows horizontally in nature. It's common. It happens every single day. Humans discovered that we could do horizontal gene transfer You've heard of GMO, genetically modified organisms. Well, let me tell you something. Every organism on the planet is GMO. Non-GMO is false advertising. There is no such thing. Every genome is constantly changing. Viruses are doing that. That's what they do. And I have to point out these orange little circles that you see surrounding the nuclei. My mom is an artist, and when I showed her a picture of this, when I was just starting out working on the germline of this animal, I showed her these really beautiful little orange things that you can see here surrounding these nuclei. And she said, "Craig, what are those?" And I said, "Mom, nobody knows." And she said, "Well, you should work on those. They're going to be important." And that is where I ended up. The Argonauts, and I'll tell you more about them in a moment, these are mediators of a genetic search mechanism. They are like the Googles of the cell. Those little perinuclear orange spheres are filled with search engine proteins that are programmed with genetic queries that are searching the information as it exits from the germ nucleus. And they are regulating everything. They are programming gene expression for the progeny, for the next generation. They're setting up the stage. This is the lineage that gives rise to that perfect new organism. It's the really unexplored territory in biology. We don't understand this process, but we're beginning to get a glimpse at the beautiful complexity here. There is so much still to learn. And what we have learned already is breathtaking, but it's just the beginning. And that's probably the most important thing that I want to say, but I do want to reinforce this notion of horizontal gene transfer because this is very important. We are entering an era of genetic medicine. Genetic medicines are medicines that are delivered from the outside back into the inside. And why is it relevant? Throughout the history of life, information has always moved horizontally. It jumps, it moves, it goes not just from your parents to the children. Information jumps across species, from one species to another. This is why the coronavirus came from bats. It's just, in a matter of 1 year, every person on the planet, probably whether you know it or not, got infected by a 30,000 nucleotide genome that mobilized all the way around the world. Throughout the history of life, organisms have been dealing with this inferno of information. And they have incredibly sophisticated internal mechanisms for handling that information. We are just beginning to understand these fundamental mechanisms that carry out and regulate the control of information that comes into us from the outside. I told you about the mitochondrial example. Well, how did plants get photosynthesis? They captured cyanobacteria and became symbionts with them. They didn't have to reinvent photosynthesis. All they had to do is capture a bacterial cell called a cyanobacteria that could already do it. And that's how plants and algae do photosynthesis. And I would argue that the genome itself, the germline, the germline, this immortal lineage, is actually capturing information, and that this is incredibly fundamental to the history of life. Being able to capture information and harness it is what makes the diversity of life on this planet possible. You think of infections as a bad thing, right? And it usually is. But it's also a way that information disseminates. And sometimes there are useful bits of information that get captured horizontally in the history of life. I don't have forever to give this talk, unfortunately. Oh, I do have to mention, one of the reasons we have such misconceptions about our ancestry, there's debates and people still don't believe in evolution in my country, probably here as well. But the Earth looked like this about 640 million years ago, and it actually, underneath those ice sheets, there was already incredibly sophisticated life. It just had to be tiny. The Earth could not harbor large animals that look like us until after this ice sheet melted. Before this one, there were many, many others. The history of life on this Earth is mostly tiny little animals, even single-celled organisms, not big things like us. We're a rather new feature. But the biology that enabled us to become big, that was already there. All the sophisticated solutions, that were necessary for us were already there. And when that ice melted, we had the Cambrian explosion and the proliferation as the oxygen levels increased of life on Earth. And I have to show you this. I don't know if you can hear this. It has a nice soundtrack. These tiny little things, these are single-celled animals. I'm just showing you that. There's actually an animal on the right, a rotifer, that's a multicellular animal. But these single-celled animals, they have a mouth, and they can do these elaborate behaviors. They have no nervous system. They are wired up to be able to behave like that as a single-celled animal. And there's a 1,000-celled animal right there that is much more closely related to the human in the middle. They make us look bad. I mean, these tiny single-celled animals, we humans just constantly overestimate. We think we're so special. We think we're the best, and we think we know everything. We don't know how those animals work. Those little guys there, they're coordinating thousands of cilia that are on their surface, these little things that they spin to control their motion through the water. They're coordinating them electrically so that they can do behaviors without a neuron. They don't have a nervous system. It's amazing. We should all be working on those. But seriously,
every creature, and you know
this is RNA.
Is made from a recipe. It comes
That's
from
DNA,
DNA,
actually.
Spelled out in chemicals, A's and C's and T's and G's, inside the famous double helix. Every creature has its own DNA, different for mice, and then for whales, and for flowers. But to go from a chemical recipe, A's, G's, and T's, to a real creature that squeaks, or soars through the air, or turns gloriously pink, that requires RNA. RNA is the thing that turns you from a chemical code to a real, pulsing, living creature. RNA builds life. That's big.
RNA, it's the software. DNA is kind of boring. It's the hardware, right? Every computer has the same hardware. But what makes a computer fun is when you can turn on a cool program and run it and do something interesting, something new. RNA is what makes the cells in our bodies different, right? They're running different programs. They all have the same DNA, but they're running different programs. And so RNA is big, really big. And I guess I put this in here just so everybody's on the same page, because not everybody knows what RNA is. And I'm going to show you, here's cartoon showing DNA wrapped up in, this is the boring stuff, and here comes what's called an RNA polymerase, and it's going to copy the information that's in this DNA. Now, this is happening right now in every person here, and that little blobby thing is very cool. It can read the DNA, and what it does is it copies the genetic information. You can see how the DNA has two strands, and it's going to make a strand that matches the strand that the polymerase is riding along. So it's transcribing the DNA into RNA. And RNA is a polymer like DNA. It's just a little bit more interesting. It's more labile. RNA can actually do chemistry. And the polymerase that you're seeing is slowed down. In this cartoon, it's slowed down because if it went at the actual speed, you would not be able to see it. It transcribes that whole strand in less than a second. It travels along at about 50 nucleotides per second. It's incredible. And again, perfectly copies the genetic information, enabling the cell to take the information that's stored in the DNA and make a copy that goes out into the cytoplasm to be made into protein. So here is the slowed down version. Imagine 50 per second, perfectly, no errors, hardly ever. And that RNA is now processed. That's called splicing. That was another Nobel Prize. It gets shipped out into the cytoplasm where it's going to find these things called ribosomes. This is like incredible biology as well. The ribosome at the heart of it has an RNA that helps it translate and read this polymer. Now you've got a linear polymer of nucleic acids, and they're being read 3 nucleotides at a time by those ribosomes to incorporate amino acids. This is a cartoon of an injection of an RNA interference molecule, which I'll tell you a little bit more about. That protein is the guided search protein, the Argonaute, that is also working in each of us right now. And it can go out, find targets, and regulate them if they match the genetic information. So the RNA, once it's expressed, can make a protein. This enzyme can destroy the message so that the cell will be able to change direction. So our cells have elaborate, sophisticated mechanisms for controlling the expression of mRNAs. And here is, I love this. I want to tell you a little bit more about this, especially the non-scientist here. This is actually really, really cool. Really cool. And I know this because I explained this to my mom and my sister and my brothers, and they always ask such good questions. This is a protein. This is a protein. You can see proteins are not just little white powders that you pour into your smoothie. Proteins are incredibly elaborate. They are a polymer. There are little, each part, that spring-like structure, that helix, those are little we call them amino acids. They're little building blocks that are assembled by that ribosome that you just saw back there a moment ago. Ribosome reads the genetic information, 3 nucleotides at a time, and incorporates one amino acid into this chain, using one type of polymer to build another polymer. But this one now has real chemical diversity, because instead of just 4 different building blocks, there are 20 different building blocks that can be used and assembled, enabling these proteins to do really elaborate things. And what this protein is doing is it's holding on to a short piece of genetic information, which we call the guide. It's a search query, and it holds it very tightly at one end. And what the protein does is it hides inside of itself most of the information in the red strand. And it presents just the first 3 nucleotides on the sort of like presents them in a pre-formed helix. And it allows this protein to interrogate information in the cell very, very rapidly at the speed of 100 million collisions per second. This protein can find matching information that matches those first three nucleotides. If those first three match perfectly, then the protein, the blue strand here is shown matching perfectly. You can see the red strand is now forming these beautiful step-like, staircase-like interactions that give the two helical turns that you see inside this protein RNA complex. This is highly precise. This is guided search. Organisms everywhere, including us, use guided search to find information. And what is so exciting about these new types of RNA medicines is that they program the search engine. They program this engine, enabling it to find and regulate targets at incredible efficiency. Oh, good. I see there's a countdown there, and I'm doing much better than I thought. I still have 9 minutes. I tend to go on and on once I get started. This is a search engine protein, and we have it. Humans have this in every cell in their body. What was so exciting about RNA interference was, at the time it was discovered, humans didn't even have search engines. Google didn't exist yet. When we stumbled upon this mechanism in C. elegans, that tiny little animal, it was very new to us. I have to say, we really hadn't anticipated the importance of a search engine, right? But if you have a lot of information, being able to rapidly search the information is absolutely critical. And life figured that out billions of years ago. This mechanism is found in every type of organism. Plants have it, fungi have it, bacteria have it. And there's a version of this called CRISPR that uses the same principle, different protein, that uses RNA search query to find and regulate and even to cleave DNA. So this is an RNA regulating enzyme, and it turns off an mRNA, but there are other search engines that also can destroy DNA. So it's really, really incredible biology, and it's programmable. And wouldn't it be great if you could make it into a medicine? There was so much hype, so much excitement about RNA interference that back in 2002, when we first realized that humans also have this mechanism, CBS News had to figure out how to explain this to the average American, which is really hard, trust me. So they came up with this very nice, very simple model that they used to explain RNAi to the audience that watches the nightly news in the US. There's the double-stranded RNA. That's what triggers the silencing. And here are the defective genes coming in from the right, left. Well, RNAi is truly incredible, but that's not quite how it works. But it is an active mechanism, right? I mean, it's an enzyme, and it can find and regulate targets. It's really, really hard to explain this to a non-scientist, and I really love trying. I do. I really love it, but I would not start with that model. I mean, that's just that is crazy strange that this would appear on the nightly news. I don't know it's incredible. I don't know. We also, I think, as P.T. Barnum said, no one has ever lost money by overestimating the intelligence of the American people, or something to that effect. Let's just say there was a lot of hype, a lot, and the drugs went into the clinic too soon. They weren't ready, and fortunately for Andy Fire and I, the hype sort of culminated in 2006, which is when the Nobel Committee decided to award the prize. So very, very good. Big pharma entered. There was a lot of investment. Companies were selling for billions of dollars. But then all of the drugs failed. They were pushed into the clinic. They weren't ready. everything failed. Big Pharma exited. Luckily, we had the prize already. But anyway, it was very, very depressing, but totally predictable. This happens all the time. It's just very, very common, sadly. But a lot of hard work was going on in the background, and during that downturn, we were able to begin our own academic efforts here at UMass, where we built an RNA Therapeutics Institute in 2008. And at the time, we felt like charlatans because there were no RNA therapeutics. So it was an aspirational name. And we recruited some great people who were working on building the chemistry so that it would be functional. And we built this building, and we're kind of off to the races. And I want to just show Anastasia Korova here, who is our first recruit, who, you know, she really demonstrated the fact that we should have all known. It takes chemistry to make these RNA molecules into drugs. And what's so beautiful about these short guide RNAs is, unlike the mRNA, which is another informational drug, it's very simple. It can be manufactured on a machine. Cost of goods under $20 a dose. And what's so nice about an informational drug is that there's two parts to any drug. There's the element of the drug we call the ionophore. That determines where the drug goes in the body. And the pharmacophore is, what does it target? You know, how does it affect regulation? But informational drugs, those two pieces are perfectly separable. Once you have a scaffold that stabilizes the RNA and enables it to deliver, let's say, to the liver, then you can very easily just change the sequence, which determines what it targets. So it's a programmable drug. And these drugs are now approved. There's several approved in humans. Through Anastasia and my colleagues at UMass, we've co-founded several companies. Believe it or not, Comanche is actually in phase 2, entering phase 2 in pregnant women. So it is so safe to inject these molecules that we can do a therapy that will hopefully allow mothers to carry their babies to term. And Stephen mentioned the importance of AI. We named this company ITHEA, and it's just getting off the ground because our slogan is, it starts with AI, right? I mean, everything starts with AI now, but just think of the power of combining information technology in the form of AI with the information technology of the cell, right? I mean, the thing that makes you healthy or sick, it's information. If you have a disease, there's an informational basis. So why not make therapies based on information? And we are. And they are amazing. And they are going to be transformative. Many, many opportunities to make really life-changing types of therapy. And I'm very optimistic, given what we now know about how to deliver these molecules, which is still a frontier. I am 100% confident that we will have cancer therapies that are transformative, that are based on informational therapy, that will basically enable us to treat cancers that are right now totally untreatable. I'm going to stop there because I want to save time. In case there are any questions, I'm happy to chat with any of you offline. These are really, really exciting times. This is just UMass. There's probably 1,000 startups right now in this space, and many of them are addressing incredibly important areas. So I think I'll just stop there. Thank you.